Really happy to introduce our second session of the Virtual Cell Development Festival Week that's put on by the Society for Mathematical Biology Cell and Developmental Biology subgroup. And so today our session is going to start off with Marc Roussel from the University of Lethbridge. So Marc, if you want to go ahead and kick us off, that'd be great. I'll stop share and then you can go ahead and share.
I think I'm good. So you can see my slides? Good.
Yes,
yeah, all right. Yeah. My title contains a bit of an apology. When I first wrote the abstract for this, I was in the process of working on a lumping analysis of this gene expression control system, and I thought for sure I was going to be able to explain some of the things we were seeing using that. And it turned out that didn't quite pan out. The lumping was almost too easy and I didn't get much insight from it. So in my group, we work on a bunch of things, but one of the things we're really interested in is the control of gene expression. And we're interested in that from a number of perspectives. And in the last few years, we've become interested in a particular class of proteins that use an iron sulfur cluster to sense nitric oxide, among other things. So this is work that was done in part by my former graduate student, Mohammed Hamidi, and our work's always funded by the Natural Sciences and Engineering Research Council of Canada, so I thought I'd put that up front. So some of the computations in particular were set up certainly by Mohammed. So this particular class of proteins we've become interested in are typically repressors. And they have this interesting property that they have iron sulfur clusters embedded in them. They are often dimeric. In fact, all the ones I know are dimeric. And they're repressors, so they bind DNA. So what you see here is a crystal structure of the one I'm going to talk about today, NSRR, bound to DNA. And if we kind of take a little closer look, we see this iron sulfur cluster stuck in this thing. And the purpose of that thing, at least in this one protein, is to sense nitric oxide. So these iron sulfur clusters, so in particular, again, in NSRR in particular, they're 4Fe4S clusters, so they're little cubes. They can react with up to 8 molecules of nitric oxide. And if you think about that, so 8 states that you can associate with reacting with nitric oxide plus the bare undamaged state. That's 9 states in total per cluster, and because they're dimers, you've got 2 of these iron sulfur clusters per dimer, and so there's 81 possible nitrosylation states, which is a bit of a pain to deal with. But worse yet, from a modeling perspective, it's not too hard actually. There's a lovely piece of software called BioNetGen that does what they call rule-based modeling. And you can kind of just put in your rules if you've got a complicated system like that for how things react. But the real problem we ran into was that there are a lot of unknowns about, for example, exactly which dimer states can bind DNA. So, if one cluster has suffered, if you want to think of it as units of damage, like in a role-playing game or something like that, if one cluster has had 2 units of damage and the other has had 4, can that bind DNA, or does it matter? Is the least damage or the most damaged cluster the thing that controls things, or is the rule more complicated? We don't know. So part of the reason we got interested in this system, quite apart from the fact that it's really cool biochemically, is that it represses the synthesis of HMP, which is in many bacteria the key nitric oxide detoxifying enzyme. So in Streptomyces collicolor, there are 2 copies of the HMP gene. And we chose this organism because there happened to be a little more data on this one than in some other bacteria. But the 2 copies are differentially sensitive to nitric oxide, which is kind of interesting. So binding of an SRR to the HMPA2 copy of the gene is abolished after reaction with 2 NOs per iron sulfur cluster, whereas HMPA1 is a little more tolerant, and the NSRR will stay bound to the HMPA1 promoter until 4 NOs have reacted with the iron sulfur cluster. Once the iron sulfur clusters have been damaged, We know there's a recycling system, but it's very poorly characterized. And there's another kind of thing that we had to kind of contend with somehow. So I want to talk a little bit about kind of how we went about thinking about this problem and the sorts of insights we wanted to derive from it eventually. So it turns out that HMP is subject to competitive substrate inhibition by nitric oxide. So nitric oxide is a substrate because the enzyme transforms it by reaction with oxygen into nitrate, but it's also a competitive inhibitor of oxygen binding. And as a result of that, models of the system, almost no matter what you assume, tend to be bistable. I don't know how much background people in this group generally have in dynamics, but by bistable, I mean that there's a range of parameters where you have two distinct stable steady states. One that you can think of as corresponding to low NO, where the system's very efficient at removing the NO because you've got lots of HMP around, and a high NO state that's essentially due to, if you get too much NO in there, it gums up the enzyme and you can't get rid of it very efficiently at that point. So we built a first model basically by waving our hands and just ignoring the fact that NSR is a dimer, so that we wouldn't have to worry about, what are the rules, how do I deal with these 81 states, and so on and so forth. So we treated it as if it was a simple protein with one iron sulfur cluster. I had done that previously with a related protein, and we just kind of went with that and just to get a sense of what this system would do. But then, because we were worried about what the dimeric nature of the repressor would do in the model, how it would change things, we then needed to build some dimeric models. But again, we don't really know what the rules are in terms of how much damage can be sustained by each iron sulfur cluster before the thing loses the ability to bind DNA. So, for example, the binding could depend on the most damaged iron sulfur cluster in a dimer, or the least damaged iron sulfur cluster, or just about anything in between. There might be a compromise, we don't really know. So we built a few variations using this BioNetGen software. And then we started drawing bifurcation diagrams, because again, so the nice thing about a bistable system is that the bistability can be very sensitive to some model details, and so it gives us a handle on how much these things really matter. So you see here, so the way to interpret these diagrams is the solid curve is a stable steady state, and the dashed curve is an unstable steady state. So what you have here is you've got a stable steady state with low NO. So this is NO along this axis as we vary one of the parameters of the model. And then there's a loss of stability. And then there's another curve up here that's stable. And between these 2 points, so if you pick a value of this parameter that's somewhere in between these two things, then you have two stable steady states, one down here and one up here. And you can see that the 3 models I picked out of the zoo of models that we tried all do pretty much exactly the same thing. Like even quantitatively, there's hardly any differences between them at the parameters we picked, most of which were inspired by, were either directly measured or were inspired by biological measurements in one way or another. One of the big things we don't know, again, because the recycling system is very poorly characterized, is how do we deal with that? So we just treated that as a simple first order reaction with a rate constant of 0.1 per second. And we assumed, because enzymes for these kinds of repair enzymes generally work that way, that they can only repair one binding site at a time. And so if you've got a dimer, you basically repair one cluster or the other and go from there. Well, when we tried changing that recycling rate constant, then we started to see differences. So you can see here the monomer models in kind of the magenta color, I guess, and the greenish color is one of the dimer models. And you can see here that the bifurcation point, so the region over which you see bistability, is quite substantially changed when you do that. It's a qualitative rather than a it's a quantitative rather than a qualitative change. That's the kind of thing in a model where there's a lot of parametric uncertainty you might not worry about so much, because you can probably tweak some parameters and resuperimpose them. But nevertheless, it clearly makes a difference there. If you had a model where many of the parameters had already been measured, then you might worry about this because it's an actual difference, but in a lot of cases, this doesn't matter so much. But the other dimer model we looked at, it does something quite different. And again, the only difference between them is the treatment of those iron sulfur clusters and the rules by which they bind the HMP genes. And what we have here is we have a stable steady state, but now instead of having a bend and a region of bistability, what we end up with, these dots here are extrema, so they're the minima and maxima of oscillating solutions. So we have this region where we have oscillating solutions, and then we go back to a stable steady state. So it's quite different than what the monomer model does. And now you've got a difference that's big enough that you start to wonder what, well, now you know that there's, you need to go back to the experimentalists and ask them to tell you exactly what those rules are, because it does make a difference. If you're curious about what's going on in some of these regions in here, the limit cycles are very complicated looking. And so every time they kind of go around one of these little, kind of sinks or whatever you call these things, I guess it's a little focus or something. You get several minima and maxima, and that's where all of this junk comes from. But it is a closed curve. It's a limit cycle. All right. So why do the models agree with fast recycling, and why do they disagree if the recycling is slower? Well, one of the things that they all have built into them, because that's what the experiments tell us, is that the non-nitrosylated NSRR binds most strongly. And as you nitrosylate these iron sulfur clusters, the binding is loosened. So again, we represented the binding reaction as simple first order reactions, repairing one cluster at a time. So if you think of alpha and beta as representing how many times they've been nitrosylated, you can go from an alpha beta state to a repaired beta state, or from an alpha beta state to an alpha repaired state. So graphically, your transition diagram based on these repair reactions looks something like this. You can go, for example, from 5,1 to 5,0 by repairing this particular cluster, or you can go from 5,1 to 1,0 because the molecule is symmetric. You can't distinguish 1,0 and 0,1. So you can go from 5,1 to 1,0 by repairing this cluster. Now, if you have really rapid recycling, then you can have two of these reactions happen, and you go back to this strongly binding state, or in general, and you tend to hover around these strongly binding states a lot more than if you have slow recycling. If you have slow recycling, what happens is you can repair one cluster, but then gets damaged again, and you start to work your way through this hierarchy away from these better binding states that are at the top of this pyramid here. So yeah, that essentially says what I just said. If that's right, then we ought to be able to look at things like, well, how much do we have of the undamaged cluster as a function, say, of the intracellular NO concentration? And again, the monomer models in this magenta color, and then in green here, we have dimer model 2. And you can see what happens as I vary the NO concentrations, that I have a lot more undamaged cluster at any given NO concentration than in the monomer model than in the dimer model. So it's exactly what I was saying. Essentially, slow recycling biases you towards having more damaged more damaged iron sulfur clusters. If you look, the thing that really matters is the promoter concentrations. So here I'm showing the concentrations of the HMPA1 and the HMPA2 clusters. The dashed lines are HMPA2, which is more sensitive, so it takes less NO for the promoter, for the HMP promoter to become cleared, because the NSRRs are losing their ability to bind DNA. But you see also that when we compare the monomer and the dimer model, the monomer model is able to sustain a higher concentration of NO before the promoter becomes cleared and it starts to synthesize HMP. So conclusion, you can sometimes ignore the dimeric state of a repressor, but not always. And of course, this would probably this would, well, not just probably, if you had a tetrameric repressor, and there's lots of those, you'd have this problem even more so. The rate of recycling, or in the case of proteins that are destroyed by an effector, removed somehow, de novo synthesis would be key. If that's high enough, you could probably get away with ignoring these complicated models with many, many states of your repressor. And so that's where we are with this at this point. Thanks.
Let's go ahead and thank Mark. Okay, are there questions? You're welcome to speak up if you have a question. You can also raise your hand if you're more comfortable with that, or put it in the chat as well. All those ways are just fine. And in the meantime, I'm gonna ask a question, Mark. So this is a new biological area for me. So I have kind of a naive question. I was wondering, you talked about like the nitric oxide states being either high or low, and I was wondering what those mean from kind of a health perspective. Like, what is it a good or a bad thing? Which one do we want?
So the reason we're really in, well, there's a bunch of reasons. I'm always tempted to go after cool systems, and then I rationalize them a posteriori. But one of the big reasons to be interested in this is that the HMP gene that's controlled by this system and by analogous proteins in other bacteria it's a virulence factor. So in other words, having that gene makes bacteria more virulent. And the reason for that is that the immune system tries to use nitric oxide to kill bacteria. There's also, in the case of Streptomyces, which is the organism we were looking at, it's a non-pathogenic bacteria. It's a soil bacteria. There's some really interesting questions about how it does some of the things it does. So you may recognize the name Streptomyces. Sounds a bit like some antibiotics you may have heard of. That's because they came from a lot of antibiotics. Streptomyces is a prodigious producer of antibiotics, many of which contain nitroso groups, so NO groups. So it makes nitric oxide, but then it also has to survive the fact that it's making nitric oxide internally, right? So it has to walk this interesting line of balancing, okay, I'm making nitric oxide so I can kill other things in the soil around me, versus I don't want to kill myself either, right? And so that control system is really interesting to us from the point of view of understanding the biology of this. I think kind of a bit of a paradigm of cells that make something toxic so they can kill other things, but still need to survive that themselves. And of course, we know a lot of cells do that essentially by just, kind of partitioning things off into a compartment. So immune cells, for example, they make hydrogen peroxide, but they make that in a separate peroxide, a separate compartment, peroxisome, right? But bacteria don't have that luxury. And so it's really interesting to think about, kind of how are they balancing all these things.
Yeah, I agree. That is a very interesting kind of dichotomy and balance. Yeah, thank you. Other questions from folks? Yes, there's a question in the chat from M18702.
Yeah, I see it.
They say, nice talk, Mark. Do you intend to search for kinds of bifurcations in the oscillation region?
I'm always very interested in dynamics, so I'm sure at some point I'll have a student look a little harder at what exactly is going on there, and not just that, but also kind of how we go from, as you vary the rate of recycling, you're going from nice, ordinary looking, bistable type S-shaped curves to these more interesting things, and how does that transition happen? So yeah, I love that kind of stuff. It's completely, I don't want to say it's completely useless. It's not biologically super exciting, but it's really interesting to dynamicists like me. And just so you know, so we're interested very much in models with delays. If you're talking about gene expression, it takes a certain time to transcribe and translate the gene. That adds to some of the complexity here, which is not taken into account in the results I have here, although we've got other models where we put the delays in, and it changes some aspects of the bifurcation curves in interesting ways. So you often get kind of, I guess, what I'd call a sort of a premature Hopf bifurcation before the saddle node bifurcation, for those of you who know about such things. And that seems to be a recurring theme, and I kind of understand why, but I wouldn't mind spending a little more time thinking about that problem too. So lots of interesting dynamics in these models.
Well, thank you, Mark. I think we'll leave any other questions for the chat. Just in the interest of time, let's give Mark another round of applause. All right, thanks so much. So Bradley, if you want to go ahead and share your screen. Our next talk is going to be by Bradley Alicea from the Open Worm Foundation, who I believe is going to be sharing a video.
All right, let me set up my, here it is.
Super.
All right. Hello, my name is Dr. Bradley Alicea, and today I'll be talking to you about the mathematics of DevoWorm, or more specifically, developmental representations. I'm presenting on behalf of the DevoWorm group, which is a participatory open source computational developmental biology group. So one of the questions we try to ask in our group is, how do we capture the multitude of interactions and state changes, which could be in terms of cell identity or other things, that occur across embryogenesis? You know, if we think about embryogenesis as a process defined by discrete and continuous dynamical models, we have a lot of options available to us. In front of you, you see a time lapse of C. elegans embryogenesis. And this is a series of images going roughly from fertilization to 27 minutes of development, roughly from fertilization to a 4-cell state. And so we can use this information in various ways to inform our modeling. One of the core things that we have worked from is this n-parameter model of embryonic development. So in the time lapse I showed you before, we have cells, we have gene expression, we have time, we have cells that are moving around the embryo. And so we can capture this in a certain number of parameters. And so here we have an embryo that I've just kind of drawn a cartoon of, and we have a number of parameters that can be stacked in various ways. So in this case, we're interested in the volume, which is the XYZ position of a cell centroid or a cell nucleus, depending on the information we have available to us. We have a time, so that we saw in the corner we had timestamp, and then we have things like migration angle, or when a cell divides, what direction does it move in? And there are other properties that we can also model, and you'll see throughout this talk what those are. So in this case, we have cell tracking data, and this is on the lower right, we have a 3-parameter point model. So what we've done is we've taken data from a primary source, and we're analyzing it as a secondary source of data. We're extracting the point cloud, and we're plotting that in 3 dimensions. So we have our XYZ representation here. On the right, we have what we call an embryo network for part of the embryo. So we've taken this point cloud, we've averaged out for specific cells, and we've built an interaction network here within sort of a 3D model of the embryo. And we've added a couple more parameters: time, division angle, the identity of the cell, and the interaction matrix, Wij. So one of the things we can do with our secondary data is we can plug it into things like networks and geometric models, but we can also plug them into these spatial data structures called cellular automata. And so in cellular automata, we're asking the question, how do we capture pattern formation in cell migration dynamics as a computational process? And so a cellular automata is a series of cells on a grid that have interaction rules, and they can contribute to transitions from something like quasi-randomness to pattern formation, or you can use things like Rule 30, which are well characterized, to look at pattern formation from randomness as well. One of the things our group has contributed to the biological application of cellular automata is this idea of nested von Neumann neighborhoods. And so von Neumann neighborhoods are a mathematical tool from cellular automata which describe the interaction between cells. And so we can scale those up so that groups of cells can interact with other groups of cells, and those can be nested in a manner that you see at the lower left-hand side of the screen. Basically, adjacent cell centroids affect the next adjacent cell centroids, so these cells are cell centroids, and they can cause global cascades throughout something like an embryo or maybe even like an organoid. And so Tom Porteges contributed this work with the rest of us in a book chapter back in 2016. Now we get to embryo networks. So embryo networks are a little bit different type of mathematical model. They're networks where you have a connectivity matrix. So you have nodes and you have edges. Those edges describe the interactions between the nodes. And so you have embryo networks. This is our paper on embryo networks. Basically, you can take that point cloud of data that we had before, and you can extract cell centroids from those and the interactions between those cell centroids using a number of different criterion, and you can get networks. This is useful for looking at things like embryogenesis and morphology in C. elegans, as well as things like the mouse blastocyst. And so this leads us to hypernetworks. So regular networks are just nodes and edges. The nodes usually have a single value, and you look at the interactions using different criteria. In hypernetworks, you have nodes that have a bunch of nodes inside of it. So it's a hypernode. Each of those nodes have their own set of connections, and you can do things like average over that group of nodes or have a variety of different outputs from a single node. And so this is something we use to model something called anastomoses, which are the connections between things. So basically what we're doing in this image is we're looking at an embryo as it's differentiating into different types of tissues. And we have different groups of cells, and they're contributing to what will become different structures, such as somatic cells, the neural connectome, the reproductive tract, and so on. And so we have these division of cell type, and we have different cells that contribute to that. Sometimes they differentiate into other types of cells, and we can build a map of that using this hypernetwork methodology. So this is an example of anastomoses in development. Anastomoses lead to things like the adult human heart, or you see examples of anastomoses in the zebrafish embryo over time. And basically, anastomoses being biological structures, we can capture those using these network anastomoses, which are defined as subgraphs of selective connectivity. And so we can also build embodied hypernetworks, which are spatiotemporal networks. As opposed to looking at something that conforms to the structure of a lineage tree, we can look at something that has a structure that's based on anatomy, and then get differences between different stages of development. So in this case, we have subcategories of cell at the 128 cell stage, and then we have new categories of cell at the 256 cell stage. And an embodied hypernetwork will tell us how those categories shift over time. So we use a number of network and tree representations in our work. We use lineage trees, which I talked about. We also use these complex networks, and we're really interested in looking at node attachment and embryo networks. So what happens when you add nodes or add cells, or cells divide in these structures? We're also interested in neural networks and perceptrons, and this is not only important in looking at connectomes and developing connectomes, but cell-cell interactions more generally. So we've done some work on lineage tree dynamics, looking at lineage trees and how those characterize the rate, recombination, and conservation of growth and form. We've done a number of papers where we've done experiments on lineage trees and reorganizing lineage trees computationally. So one of the consequences of working with lineage trees is that we can move towards other theoretical concepts. And one of those being cellular decision-making around something like game theory. And so we've introduced something called embryogenetic game theory. This was Rob Stone and colleagues looked at this concept of game theory as applied to cell divisions in an embryo. So we introduced this concept of first mover dynamics, which is something that is where the first division that occurs sort of sets the cadence for the rest of cell division in an embryo. So first mover dynamics is something from the economics literature, and it's defined as starting with a slight asymmetry and following that out. So the first move constrains the second move, constrains the third move, and so forth. There are different rules. You can start with different initial conditions, and it leads to a larger consequence. So we can model this as a series of cell divisions where we model, say we start with a rule where the larger cell in the two-cell state moves or divides first. You get these cells that come from that division. They take up their space in the embryo, and the second mover then is constrained in terms of space. So this defines sort of the origins of asymmetry and allows us to find those origins of asymmetry, but doing it in a way that's consistent with a lineage tree, that gives us a cellular decision tree and can lead to other kinds of theoretical constructs, such as a differentiation tree. We explored this in a number of embryogenetic contexts in this paper from 2016. This is an example of embryogenetic game theory from the first move to the 8th move, and you can see the dynamics. And if we started with a different initial condition, we'd get different results. So finally, in the future, we'd like to model curvatures and compliant surfaces. So we think of an embryo as like a complex geometry with different sorts of material properties. So we can model things like mini-gut organoids, or model embryos as soft active materials where we have myosin and cross-linkers and actin filaments all represented. So this represents a departure, what we were talking about earlier, with singular cell centroids, or movements of the cells themselves. And so we're interested in something called tensegrity, which is how the cell is hyperstable, or how the embryo is hyperstable in terms of its structural integrity. And so those are all very interesting questions, but we first have to think about mathematical models to sort of approximate those things in a computer or just in a model that we can then generalize from. So we've looked at a number of things. So here we have this, we go back to our original model of XYZ dash dash dash, and we can actually add in aspects of the position that are amenable to modeling curvatures and compliances, so shapes and surfaces. So this is a recasting of our original multi-parameter model, where we have the ability to integrate position over curvature and integrate position for movement in a soft material. And so this, we can modify our data structures to represent continual shape formation on a three-dimensional surface. And again, this unlocks a number of theoretical concepts that we can explore. One really nice example of this type of modeling and representation comes from this paper, Developmental Incongruity is a Dynamical Representation of Heterochromy. And so if you go to this paper, you'll see a lot of really cool modeling examples. But in this example, what we're doing here is we're building a model of the embryo that's explicitly spherical. And we're taking that sphericity of the cells and the tissues and the differentiation tree that emerges from different tissues differentiating at different times. And we're mapping it to the outputs of a gene regulatory network architecture. And we're visualizing this as what we call an expression tree. So these different colored blobs are tissue at different levels of the tree. So as the differentiation tree unfolds, like a lineage tree, you get the formation of different tissues at different times, and they descend from other precursor tissues and cell types. So we can see this mathematical model here. You have the depth of the tree, the angle of differentiation, the gene factors acting in cis. The depth of the tree is where it's acting in trans, and then the blob volume itself, which are these little colored blobs, and their size. So the idea is that their size are the number of cells in that tissue, and the depth is the number of branches in the tree. So thank you for your attention. I'd like to draw your attention to something I didn't talk about explicitly, but we're on Hugging Face and GitHub with our DevaLearn platform. So DevaLearn is a machine learning platform that we've been working on to analyze different aspects of developmental data and build developmental models. So we've got we work with a lot of neural network techniques. We work with graph neural networks, recurrent neural networks, and we're interested in analyzing primary data and extracting theoretical and mathematical insights. And also, this is the 10th year anniversary of our group, so check out our YouTube channel. We have our weekly meetings there, as well as some other talks and goodies. So thank you for your attention.
Thanks so much, Bradley. Let's give them a round of applause. Again, if folks have questions, you can just go ahead and start talking. You can raise your hand if you prefer, or you can put it in the chat within Zoom.
Do you mind if I jump in?
Go for it, Marc.
So, a really interesting talk. I like, kind of thinking about this on different styles of models, because of course they're useful for different things. But one of the things I didn't quite yet, and I'm not, not a criticism, I'm just trying to understand kind of how far you can go with some of the ideas you presented. Is to what extent are you able to model complex shapes with some of these techniques you were talking about?
Well, it depends on the technique. So with the cellular automata, you can build three-dimensional models, and you can model shape in different ways. Some of the methods, not so much, because they're sort of, dependent on like the discrete models, they're dependent on discrete steps that you're modeling. So I didn't present all the models that we use. We have a lot of them. And, one of the things we haven't done is really integrate them in a way that would really capture, like a three-dimensional context and be able to model all the changes. So, we have some models that kind of model one aspect of development, another model that models another aspect of development. So if you picked something that was very amenable to complex shapes, like the cellular automata, that would do adequately, but you'd be missing out on some of the other things that we're doing. So maybe one of our future goals is to bring these models or align them more closely. So that's a challenge, but also, capturing that three-dimensional shape as well. That's really reliant on our input data. So we have a lot of cell tracking data that's available, but that only goes so far. You can only model, say, for example, maybe the membranes or the cell centroids. And so being able to capture, you really have to approximate that shape in different ways. So.
Thanks, Mark. Other questions?
I can ask a question.
Oh, yeah. Do you want to go ahead first? There's also a question in the chat.
Or
Go for it,
yeah, go for it with the Leah first.
Okay. Bradley, Leah Edelstein-Cushet has a question. She's asking what the nodes and edges represent in the networks.
So in the embryo networks, they're generally cell centroids, and then there's some relationship between them. It could be distance, it could be cell-cell signaling. It could even be like gap junctions or other types of properties. So you're really abstracting away, a lot of details to get to these kind of models. But you can actually use a number of stand-ins for your nodes. So you could use a cell centroid, you could use a cell nucleus, if you have a nuclear marker. You could use other types of, criterion depending on what kind of input data that you have. So it really depends. But most of the examples that I showed you were basically cell centroids and then some relational criterion.
Super. And Maria, did you have a question?
Yeah, great talk and very interesting. I have I guess more of a general question, because you keep talking about these different models. Are they all on the same platform, or are they separate? How are you combining all of these different efforts into this one system? I guess what I'm missing.
Oh, well, I guess we're not really integrating them. Like I said before, not really integrating them in a meaningful way. Our history of our group, we've had people asking different types of questions, interested in different types of things. We've kind of proliferated models in different directions. So like, some of them are, very tied to secondary data. Some of them are very tied to theoretical questions. Some are very tied to like machine learning imperatives, things we need to do for that. So they're not, that's again a problem, because they're kind of going in different directions sometimes. And, one of the problems we've had is to try to find like a unified way to like, use the different advantages of each model to really pull them together. And, that's a problem too in modeling like development, is that there's so many things going on, so many things you can, like details that you can include or exclude, that, depending on your question, you almost end up in a different place. And, you can build integrated models, but we haven't done that. We haven't taken that approach.
Thank you.
Sure. All right. Well, let's go ahead and thank Bradley again. Just in the interest of time, we'll leave any other questions to the chat. Okay. And so then we have one more talk in the kind of research portion of today's session, and then we have a professional development panel. So we're excited for our next speaker. Elizabeth, are you ready? If you want to start sharing your slides. Our next speaker is Elizabeth Trofimenkoff, who's our second speaker from the University of Lethbridge. And so take it away, Elizabeth. You're our research finale today. Let's see, I think you may be on mute, Elizabeth.
I am definitely muted. My bad.
There you go. All right, very
It's
good.
been a while since I've done that.
Go for it, Elizabeth. Happens to the best of us.
Awesome. Thank you so much for the introduction. Thank you for having me. I'm today going to be presenting on some of my master's work, but before I get into any of the content, I just want to point out that this research is actually one of 5 studies working on an overall project of developing a new chemotherapy agent for folks who are suffering from glioblastoma. So you can see many of us are from the University of Lethbridge, but we also have a colleague at Queen's University Belfast, Dr. Jafarnajad, out in the UK. And of course, this is all funded by the New Frontiers in Research Fund. So this is where I go back and get my laser pointer. Perfect. So glioblastoma is a deadly brain cancer that affects 4 in every 100,000 Canadians each year. It has a median survival of about 15 months and a 95% mortality rate within 5 years. Now, unfortunately, only about 50% of patients respond to standard of care therapies. And there are other therapies in clinical trial and development stages, but no effective cure has come around. Before I get into anything too much further, I do want to talk a little bit about this process called apoptosis. So it's a form of programmed cell death. It's a very important process in development in general. For example, it's the reason we have identifiable fingers and toes. So along a cell cycle, it may come across some kind of stress, whether that be oxidative, nutrient stress, heat, you name it. And then it has kind of one of 2 things that it can do. It can survive and overcome that stress, or it will die via many different things, but one of these things being apoptosis. There's these interesting proteins called inhibitors of apoptosis, or IAPs, and these will basically force the cell to survive because they inhibit this apoptotic pathway. And the one that I was primarily interested in is ZIAP, so the X-linked inhibitor of apoptosis. This protein interferes with the caspase cascade in the apoptotic pathway. It's overexpressed under hypoxic stress conditions, which we see in glioblastoma, and it can be translated using a non-canonical pathway facilitated by an internal ribosome entry site, or an iris, in its 5' UTR. So we're primarily focused on the translation initiation of this protein. There are 2 ways that this protein can be translated: using a normal canonical pathway, like many other proteins, where EIF2 is the protein responsible for delivering the initiator tRNA. However, under stress conditions such as hypoxia that we observe in glioblastoma, the alpha subunit of EIF2 is phosphorylated, and so it can no longer bring the initiator tRNA to the ribosomal complex. And that would, potentially stop the synthesis of ZIAP. However, it can be translated using a non-canonical pathway as well, where this other protein, EIF5B, can deliver the initiator tRNA under stress. This is in one specific form of non-canonical translation initiation, particularly for iris-containing mRNAs like we see in ZIAP. Now, there is one step that is present between both of these initiation methods, and that's the bridge between initiation and elongation, and EIF5B needs to dissociate in order to make that bridge. There's a few things that we need to consider when we're modeling dynamic systems, especially for gene regulation, and Marc had already talked about this a little bit earlier, and that's this concept of delays. And this because no process happens instantaneously, but particularly when we're looking at gene expression, that takes certain amounts of time in order for an mRNA to be synthesized or a protein to be synthesized, but it also takes a little bit of time for, say, a promoter region on a DNA to become available again, or the ribosome binding site to become available again, or in this case, the iris to become available again for another ribosome to bind. And so there are two kinds of delays that we can consider. Tau1, which would be a clearance delay, and that's the time it takes for that promoter or ribosome binding site to become available again. And Tau2, which would be a synthesis delay, like how long it takes to make the mRNA or the protein. Now this just as a schematic here, so if we consider this red zone here to be the iris, what I mean is it takes some time units, tau, in order for that ribosome to clear that iris so that another ribosome can bind. If another ribosome is trying to come and bind onto this iris while it's occluded, you'll notice that it can't bind because quite literally there's no space for it to go. So we have to wait in order for that iris to become available again. What does that mean mathematically? Well, like I said, this is the bridging step actually between initiation and elongation, where EIF5B must dissociate. But then it's going to take some time for that ribosome to actually move off of the iris in this case as well. So the rate at which that iris becomes available would be K7 times whatever the concentration of the RIB5B complex in this case has to be. But when we're looking at creating a mathematical model, one of the things that we have to consider is the overall conservation of the mRNA that's in the cell. And so we represented that by looking at the concentration of the iris in this case. And mathematically, we need to incorporate a term that looks like this, where this would be the amount or the concentration of the mRNA or the iris that is occluded by that ribosome that's not available for another ribosome to come and bind. I had a few research questions, but before we get into that, just as a recap, glioblastoma, extremely aggressive and deadly. Only 50% of patients respond to standard of care therapy, and there's no effective cure. There are other things in place, however. So my idea was to, well, not necessarily my idea, but my research process was to model the non-canonical pathway and determine a few plausible therapeutic targets. Then I wanted to look at the effects of different potential inhibitors on the non-canonical translation initiation pathway, but also consider the effects that it may have on canonical translation initiation, because we want to have minimal toxicity to the patient overall. And then, of course, compare these properties to identify which one of these is the best potential inhibitor moving forward. So 3 main questions. What's the concentration of EIF5B necessary to inhibit the non-canonical mechanism? What kinds of inhibitors could we propose? And what properties of these inhibitors are necessary so we do limit the toxicity of the patient? Before I get into any of the results, I do want to point out that, as always, we need to consider the parameters of the model. So we did a local relative sensitivity analysis and tested a few different parameters. So the delay time, the EIF5B cellular concentration, the mRNA concentration, and all of the unknown rate constants. And it turns out that the only parameter that the system was sensitive to was the mRNA concentration, which makes a little bit of sense, considering that would be the limiting reagent in the situation. It has the lowest concentration out of all of the things we're looking at. And I should say that the ZIAP mRNA is sometimes about 30 times higher in glioblastoma cells than a normal cell. And so I did test it at a 30 times higher concentration as well. And we still got the same results, that the system was only sensitive to the mRNA concentration. In fact, it didn't change the sensitivities of any of the other parameters I was looking at either. So first things first, let's talk about what concentration of EIF5B we need to actually inhibit this pathway, or to perhaps a more medical perspective, what is the effective concentration of EIF5B that we need to decrease the number of ribosomes, in this case by 50%, so an IC50. And it turns out that the number of ribosomes saturate around 700, so we would want something, around 350 ribosomal complexes, and it turns out that we would need a concentration of less than 10 to the minus 3 micromolar, 1 nanomolar in this case. So that is like quite a decrease from their normal cellular concentration in which we observe this saturated amount of complexes. Now, there are a couple of modes of inhibition that we considered. So the first one, probably the most obvious, is let's just inhibit EIF5B directly. The other one is like, we're inhibiting EIF5B, but in its ternary complex. So after it's bound to the initiator tRNA, maybe we can stop it at that level so that it gets a little bit gunked up and it can't move forward with the initiation. We could also think about inhibiting, instead of the ternary complex, inhibiting the interface that the ternary complex would bind to. So it's kind of just like the other side of that complex. And then we could also think about perhaps targeting the complex that's before we actually have a competent ribosome to move on to elongation. So we're basically stopping it at the initiation step. Now, I will point out that this complex right here, if you remember, is present in both pathways, right? So we would be careful when it comes to this complex here, which we'll talk about a little bit later. It turns out that if we look at the EIF5B, the pre-initiation complex, and the ternary complex inhibitors, they all look very similar. And we can vary the inhibitor concentration and the KD of this theoretical inhibitor and look at how that will affect the number of ribosomes able to translate the ZIAP mRNA. And we get this lovely 3D surface plot, which we can, of course, translate into a contour plot, which we see here on the right. And if we're looking at IC50 values, everything along this second line right here that I'm highlighting with my laser pointer, every single one of those values on this line will give you 350 complexes. So if we arbitrarily choose a 1 nanomolar inhibitor, which is a pretty good inhibitor, it turns out that the concentration of inhibitor that we would need is about 2 micromolar. Now it's a little bit different for the for this like RIB5B complex. You see that there's a much larger area of inhibition, which sounds really promising because we need a very small concentration of inhibitor, only 0.003 if we compare that to the same 1 nanomolar inhibitor. But again, we have to be hesitant because this complex is present in both the canonical and non-canonical pathways. And so naturally, the next step was to look at a couple of canonically translated proteins, such as type 1 collagen and beta-actin, 2 very important maintenance proteins, maintenance and structural proteins. And under normal healthy cell conditions, these are things that we don't want to impact, right? If we're giving a chemotherapy to a patient, the last thing we want to do is impact the healthy cells. And it looked promising here when we were talking about the RIB5B complex and that concentration. However, we notice it's a very similar concentration to impact canonically translated proteins as well. Interestingly, even if we have poor specificity of the inhibitors, we see that the concentration required to limit the number of ribosomes by half is much higher. So it was around 5 micromolar in the non-canonical ZIAP case, and here it's 80 and 100. So it's much higher of a concentration we need to impact this canonical pathway, which is very promising moving forward. So what concentration of EIF5B is necessary to inhibit the mechanism? Well, we said that was less than 1 nanomolar or 10 to the minus 3 micromolar. What kinds of inhibitors are required to inhibit this pathway? I'm going to throw you for a loop a little bit and say that perhaps the EIF5B inhibitor is a good option. And the reason for that is because there is some experimental evidence saying that EIF5B is not essential for canonical translation initiation. In fact, cells can still go through and create proteins through canonical methods with a knocked down concentration of EIF5B in the cell. The cells are still viable. Now, of course, probably the safest one would be to have a ternary complex inhibitor because that ternary complex is not present at all in a canonical pathway. And finally, what properties of the inhibitors were necessary? Well, we discussed a number of IC50 values throughout here as well. I would like to thank Professor Roussel, who's here as well, and my other co-supervisor, Professor Thakur at the University of Lethbridge. Of course, the Virtual Cell Development Festival Week organizers for having me, all of the lab members, and my cats for all the emotional support. And I would also like to thank the New Frontiers and Research Fund for funding this research.
let's go ahead and give Elizabeth a round of applause for that nice talk.
Yeah, it was really good on my 15-minute mark. Nice.
That was a very nice talk, and I appreciate the personally appreciate the shout out to cats.
Yes.
Are there questions from the audience, either in the chat, or you can raise your hand, or you can just start talking if you have a question. And I will get us started off. But first of all, I really appreciated that you could see the different concentrations that were needed for inhibition and the different approaches. And you had those 4 different kind of methods for inhibition that you were talking about. Are you considering doing combinations of them in the future?
Like if we were to combine a direct EIF5B inhibitor and a ternary complex inhibitor
Yeah,
together.
is that possible? Or
That's very interesting. Yeah, it's mathematically possible, of course. Yeah, that wasn't something I considered on that project, but it would be really interesting moving forward, particularly because at least what some of the clinical trials have been saying is that combined chemotherapies tend to have a better effect. So, I mean, it depends on what chemotherapies you're talking about, but patients seem to have a bit of a better prognosis. So,
Thank you. Maria?
Great talk. So this is a new field to me. So I guess wanted to sort of understand some things. So when you said you had that tau, which sort of covered your time, you weren't talking about it translated, like you didn't seem to be interested whether it was mRNA or protein, and that sort of like struck me as interesting, because a lot of the times and we can talk about this later, but a lot of times when I propose, like, you know, how things are translated and I'm only considering the mRNA, everybody's like, well, you're missing the whole splicing and all these other events. And I find it really interesting that that didn't seem to matter, or wasn't as important in your model.
Yeah, that you're absolutely right. Like the splicing time it's funny you mentioned that. That's what I'm working on at the moment for my PhD project. So, yeah, at this point with this model, I was only focusing on translation initiation. And so this is after splicing has occurred. We actually have a properly synthesized mRNA that is ready to basically go down the chute and get translated. So I've like kind of like started time at when the mRNA is able to be translated, I guess. And the only delay that we would have to take into consideration at that point would be the clearance delay, because I'm only looking at initiation. If we expand the model later, there are some elongation models and stuff as well. But then we would, of course, have to consider the time it would take to translate the entire protein at that point. But yeah, I'm just like very like binoculars on looking at just the initiation step.
And I guess my question was also more about, let's say you're initiating, I know you're saying translation, but to go to a protein, but is it the same as, do you think it'd be the same as initiating a transcription to go to an mRNA, or that's a different process, you think?
Well, the principle of a clearance delay is similar, right? Because then you have, but the times will of course differ, right? It's how long the polymerase takes to clear the promoter region, how long it takes to assemble, and then, you know, clear the promoter region kind of idea, right? And then it's similar when you're looking at translation. It's how long it takes for the ribosome to assemble and then move off of either the ribosome binding site or the iris in this case. It's interesting in this specific scenario too, because there is an iris, and I didn't talk about that, but the iris is it's a secondary structure in the RNA. And so it's possible that it will be deformed during the ribosome assembly process, and it will take some time to actually reform that secondary structure as well. Now, that's like kind of getting into the weeds a little bit, but there's lots of things that kind of come into play when you're looking at that clearance delay too.
Interesting. Thank you.
Thanks for the questions.
Super. So I think in the interest of time, we'll go ahead and stop with our research talks there. Let's thank Elizabeth again, and also Mark and Bradley for those excellent talks. Super. All right. So I don't know if everybody wants to grab dessert or a drink or a coffee or something, and we'll go ahead and get ready for our panel. So we have several panelists who are going to be joining us for a career panel on industry positions, and we'll start in about a minute or two. Hi, Robyn. Hi, Stephen.
Hey, how are you?
Good to see you.
Yeah, you too.
So I'm just gonna grab some water as well.
It's later for you guys right now, right? You're on the East Coast. Or are you Midland?
It's 8 p.m. over here. What time is it in Toronto, Maria?
It's the same, 8.
Okay.
I'm still on California time though, because I was in California until Saturday morning. So-
Okay. Yeah. I got back from the UK on Saturday evening, so I'm like, the
I
journey
don't know what
back
time it
was
is for you.
okay. Like
Yeah.
the jet lag, not really much jet lag. I'm maybe a little tired, but it's definitely worse on the way over. So yeah.
And welcome everyone. We're going to get started in just a minute or two. You were in Leiden, right, Robyn?
No, I was just in Cambridge
Oh, okay.
and
I thought
Scotland
it was
for
Leiden.
a
Okay,
few days.
gotcha.
Yeah. Busy time. How was your trip to California?
Very good.
Good. Yeah.
Veronica was also there, who's in the room,
okay.
or at least.
Nice. We have nice weather right now, so yeah.
All right. Super. Well, I think we will get started. So we have two panelists who are joining us today. You can see, I don't know if, Maria, is it possible to kind of spotlight them, do you think, or in Zoom?
I don't know
It
how
may
to
or
do
may
it.
not be, but we'll
Yeah.
have Robyn Shuttleworth kind of wave her hand for folks to see, who's joining us from Altos Labs, and Stephen Tenholder, if you want to wave your hand, who has experience with startups and all sorts of things. So we're really excited for both of them to be sharing their experience and talking about their career trajectories, and they've both participated in lots of things and will be able to provide some exciting advice. All right, so what we figured is we'd have them both introduce themselves, and then I have questions prepared, but we want this to be informal and a conversation, so you are welcome if you want to use your camera, but there's also no pressure if you prefer not to. You can raise your hand and ask questions. You can speak up and ask questions. You can put them into the chat window, and we'll also get to them there. So there's lots of options, but just to get us started, I'm going to ask Robyn and then Stephen to introduce yourself, tell us a little bit about your career trajectory and your current role. So Robyn, if you want to go ahead first, that would be great.
Thank you. Yeah, so I'm Robyn. I completed my PhD in applied mathematics in 2019. My PhD was in cancer biology, so I worked on using a multi-scale model to describe the tumor morphology and how it interacts with its environment, the extracellular matrix. And I did that in Scotland. I then moved to Saskatoon in Canada for my postdoc, and I moved into a cryobiology lab. So I was looking at trying to optimize some of the cryopreservation protocols there using PDE models. And then just in November 2022, I abruptly moved into industry, I'll say. I had my heart set on academia, but I moved to industry into Altos Labs, and I now work on computational models of aging and cellular reprogramming.
Thanks,
Thank you.
Robyn. Stephen, if you want to go ahead. Sorry.
All right, great, cool. I'm Stephen. My North Star since grade 10 has been human longevity. So kind of coming full circle with Robyn there on Altos Labs. Went to Waterloo, did iGEM for a number of years. I'm sure you're familiar, synthetic biology.
I did too.
Yeah, really, that's cool. On the University of Waterloo team, worked with a number of different organisms and systems, CRISPR and Arabidopsis, et cetera. And I thought I would be an academic, but I had an idea for cryopreservation, surprisingly. If we're going to get better 3D bioprinting, tissue engineering, obviously Shinya Yamanaka had happened, iPSCs, it seemed smart that everybody should be preserving their young cells. Started the company Acorn Biolabs. So went through, I really thought it would be very quick, like a Kickstarter kind of thing, 10,000 people from Silicon Valley putting their cells in a freezer and call it a day, and I go on and do my PhD, but it turned into much more than that. Went through kind of Y Combinator, Creative Destruction Lab, a bunch of different accelerators and incubators, moved from Waterloo to Toronto, raised millions of dollars, and launched product. So that company is still going. About 3 years ago, I took a step back, and then about a year ago, I was able to sell about a portion of my stake in a secondary, and now I am on sabbatical, and I'm deeply exploring LLMs. So could we use them to understand the language of life? And that can mean a lot of things, and there's a lot to find out, the strengths and the weaknesses, but that's what I'm spending my day-to-day doing now.
Super. Thanks, Robyn and Stephen. So I can get started on questions, but if there are questions from the audience before that, anybody have questions to get us started beforehand? Okay, then well, let's see. So first, I wanted to ask about your kind of your day-to-day, like what in if you've had multiple roles, you can pick out one of those roles, but what do you think is your kind of day-to-day, and how is that different or similar from your experience in graduate school in your current position?
Robyn, you want to start?
Yeah, sure. So I will say in industry, I have a lot more meetings every day. I didn't used to have so many. So we have within the company, you end up meeting with lots of different people who are there working, and you collaborate with each other. So I have a lot more meetings, I would say. And the rest of my time, I tend to spend individual working, I'll say. So I still do a lot of coding. My base is still MATLAB. So I spend a lot of my time doing that. So generally, we just have meetings, maybe some seminars that we have. We sometimes have speakers, and just, collaborating with people, and then individual work. So it's very similar to a postdoc, just like I said, more meetings. Other than that, it's very similar.
That's actually not too dissimilar to mine as well. I'm doing a lot of networking. I see a lot of programmers in Silicon Valley that are becoming demystified with creating another version of Snapchat, and they want to turn their talents toward greater things. And so my job is to sort of sell biology to them. And so quite often I'm going to these events of startup founder type people, but who have really good technical software development skills. And I'm trying to go deep into the sort of machine learning people. So networking and pitching biology is a lot of my time, and then programming. I did not have a formal background in programming, but thanks to GPT 3.5 and 4, I've learned a lot recently. And I've been surprisingly successful at doing things like fine tuning and even some more architectural things with large language models. So I'd say, half of my time is at a coffee shop doing programming, and half of my time is networking and meeting new people and setting up foundations for a next startup. And then also working with Maria at PhD. It's really fun work. And thanks to everybody here, by the way, for networking. I'm learning a lot from the state of the art from you guys.
Super. I feel like Stephen, that half my time is emails. It's good you've got half time research and meetings. So Robyn, I wanted to ask you, so you're talking about the meetings that you have, and you touched on also on software. So when you're working on meetings in your position, do you feel like are they mainly working meetings, or is it more like big picture ideas? Do you ever program in pairs? Is that something that happens in your company?
Yeah, I would say more often than not, it is more like big ideas or seminars so to learn. So as I said, I came from cancer biology to cryopreservation, then to aging and reprogramming. And so every field I've taken is a new field. So I have a lot of learning to do. And I think that's really why I enjoy switching fields so much, as I enjoy learning. And I feel like I can take things from previous fields and apply them into different fields. It's just the nature of biology. So I think I spend a lot of my meetings and seminars learning from my colleagues. And I like it that way. I very occasionally will have like working meetings where I code with people, but generally it's a more learning environment for me right now. So yeah.
Okay, we have a question from an audience member that we'll take in just a moment. But Stephen, I did want to ask, because you talked about meeting with all sorts of different folks, what do you feel like what is the degree background and kind of fields that the people are coming from in your position and in your career?
Yeah, interesting. Silicon Valley, especially the software people, have this sort of like, I don't know if you guys have seen the movie The Social Network, but there's a scene where Mark Zuckerberg, he has a lot of traction. And so he goes to a venture capitalist in flip-flops and a hoodie, and he has a card that says, I'm CEO, B word. And I think they kind of have that attitude where it's like, I'm skilled and I have a good set of projects on GitHub, and that's enough as credibility and the places that I've worked. So they typically have this sort of like non-academic perspective, or a lot of them do anyway. Or some of them are jaded, or some of them are cynical. I'm not quite that, and I do think that there is an important role for academia in the world, and especially in the world of biology as it meets software. But I definitely have a lot of my sort of like network and connections in that attitude of the world. And I mean, one nice thing about it actually is that the barrier to entry is very, very low. Any of us here, if you showed up and you just started talking about your mathematical models and the first principles behind them and how we're implementing them in our own versions of software and modeling, they'll just start talking to you in the trenches immediately, in the weeds, and they're quite engaged that way. So low barrier to entry and very open environment. When you kind of, one person, you meet 5 more, and then 50 pretty quickly.
I also meant in terms of, do you feel like it's mostly computer scientists, or it's physicists, or it's biologists, or it's mathematicians, or like what are the kind of majors? That's what I meant, like the kind of the departments that they're coming from in that sense.
Gotcha. Yeah, software. So different universities say it differently, but like computer science and software engineering, that kind of thing is the number one. But there's a few people from physics that have moved over to biology as well.
Super. All right. So we have a question that it would be great to hear from both of you on, from Kyle Nguyen in the chat. So they've asked, when did you start realizing that you wanted to work in industry rather than academia?
So I actually never even had the realization. I was accidentally flung into industry. So I'd been in my postdoc for just over 2 years, and I'm quite active on Twitter. So it's my favorite platform. We see lots of research and things on there. So I'm always on there. And I'd seen a lot of just chatter about, salaries in academia, how difficult it was to get a job in academia, applying for grants and things was very stressful. So I think I kind of had the idea in my back of my head that I wasn't 100% sure if that was still the path I wanted to go down. And I really wanted to speak to people who had been in academia and who had left to go to industry, just to get the perspective and see why they might have done something like that, if I might be considering it. And so, yeah, I think it was really a good chunk into my postdoc. I had a 5-year postdoc secured, and I left after 2.5. So I think at that point, I would have been searching for jobs in academia anyway, and I just kind of stumbled upon my position now, actually through Twitter. And I really wasn't looking for a job at that time. I had reached out to someone who I didn't know, I had never met, never spoken to, and I knew they'd moved from academia to industry, and I just wanted to have a conversation. And one thing led to another, and I had an interview, and then I joined. So it wasn't like an instant thing that I thought about. It was just kind of niggling at me. I'd fully prepared for academia. I was teaching, I was building courses during my postdoc. And these were all unpaid things I was doing, but I was so keen to stay in academia that I was trying to build my CV for that. And yeah, all of a sudden things just kind of tipped on their head and I moved to industry. So yeah.
Second on Twitter on that, I feel like LinkedIn is nice for people to have their resumes and to speak kind of formally, whereas Twitter is, it sifts through to really good insight, and you really only network with the people who are very transparently sharing great insights and data and information. And I mean, obviously drama, but like often drama is what's interesting because there's some substance to what somebody's discovered, and that leads to this and to that, and it's going to change the world in this way or that way. Like even Altos Labs itself, of course, has a whole PR thing going on with Jeff Bezos, but that's part of probably the excitement of even just kind of working there. Yeah, so I met Maria through Twitter as well, and I really like engaging on Twitter. So I'd encourage everybody to do more of that and just share random insights and stuff. And let's get the science community back on Twitter.
Super. So we have another question in the chat. This is from Allison Cruikshank. They've asked, at the beginning of your careers, were there opportunities for training and skill development in mathematical methods and tools within your companies? So I think that's like when you were first starting in your industry positions. I don't know if Robyn, you want to get started?
Yeah, I've been going first, so we'll continue. Is that cool? Okay. Yeah, so I actually decided that I mainly focused on PDE and ODE models during my postdoc and PhD, and I decided when I joined Altos I wanted to do agent-based modeling. I was familiar with it, hadn't done it before, so I decided that's what I'm going to focus on, and now I have 3 or 4 different models, ABMs, up and running. So I did definitely have the opportunity to explore a different kind of area of mathematics and train myself in that. And we are encouraged every year to, focus on something that we might not be familiar with, and then we can learn. We have a lot of colleagues here who are just experts at what they do. So we have tons of people to learn from, and we're definitely encouraged to kind of pick something and make that our goal for that year to kind of develop skills. So maybe they're shelled something like Python or maybe machine learning techniques. But yeah, there's definitely lots of opportunity to keep learning. And I think that's the most important thing for me. So
For me, it was definitely very different. In the startup world, let's say you get some seed funding and it's a group of 5 of you and you just show up at the office and then you look at each other and you're like, all right, what do we do next? Exactly. You've got a vision and you've got some ideas of direction, but then exactly precisely what are the things to work on and not work on? So you're learning all the time. And not only that, you're learning what to prioritize and how to prioritize it all the time. And you're answering basically to yourselves and your investors as opposed to your grant funding body and your PI. Yeah, I think learning initiative was a big one, learning what not to do, where to cut dead ends off, and learning, new skills as they came up. Another nice thing about the startup world is I think people are quite open to talking to you, and there isn't as much maybe red tape or reasons not to talk to people. You kind of just reach out, and somebody that's willing to talk to you, you can learn skills from them. And you can offer them equity depending on whether they like that kind of thing. You can say, hey, do you want 0.5%? And you keep talking to us every week about your skills and knowledge, and you update our team on their skills and knowledge. For Acorn, we didn't have to do as much mathematical kind of development and software development in the first few years. And then after that, we did use AI to do cell sorting. So that was great. And actually, I hired somebody whose job I told him it was to teach me for like 2 hours a week. So that was pretty great too. It was, I think, a creative way to learn as well.
Sure. Thank you both. So in terms of kind of piggybacking on this idea of skills development, when you think back to your time in graduate school, or if you had a postdoc, your postdoc experience, what do you feel like were skills that were like particularly useful for making the transition into your careers that you learned in those programs, and maybe places where you feel like there are gaps where we need to better prepare students for these types of positions?
So I think this is more of a personal goal, I guess, or skill. Is presentation skills are huge. And I think that you're not always given the opportunity to do that as much. Definitely I wasn't during my PhD. I really had to force for attending conferences. It wasn't something that was terribly encouraged, so I really pushed to attend lots of conferences. And especially during my postdoc and during COVID, doing conferences online was I was just trying like every couple of months to get some presentations in. And I think learning how to explain your work in layman's terms is a massive skill that you need if you're moving into industry, because you need to be able to speak to people from all different fields and be able to really like give that information over. So I think that's something that we're not necessarily given the opportunity to do during our graduate studies, but I definitely think it's something we should try personally to really get out there and do that for ourselves. So.
Yeah, I double down on that 100%. What else though? Yeah, I mean, at Waterloo there was a science and business degree that you could take, and there was a number of students that were in that, and I felt like that was a good initiative for them to take, especially Waterloo being kind of very entrepreneurial as a university. But even there, it maybe like first principles understanding of like money in science in general, right? Like it is very similar. Like a PI is the CEO of their group, and they go to the grant funding bodies, which is a combination of government and obviously industry sometimes. But I feel like maybe just understanding value and like value chains, it would be a great world where basic research could be done, and you don't really have to think about that. Like 95% of your thinking can just be on the basic research, but we're not quite there yet. And I think if people understood sort of like value capture better as scientists, then the transition from academia to doing a startup instead of necessarily becoming a professor and going for grants would maybe be more fluid. I mean, I'm benefiting from it because I get to go to a bunch of academics and transition for them or help, or I think that's kind of the niche I'm unfortunately now going into. I'd rather do more science, but I'd like to get more scientists thinking about value capture, however not fun that sounds. Yeah.
Thanks, Robyn and Stephen. Okay, we have another question in the chat from Kyle Nguyen, and these questions are all great, so keep them coming if you have more questions. So this one is, there were a lot of layoff announcements from biotech and pharmaceutical companies in the past year. Do you have any advice for new PhD graduates who will have to compete with other candidates with more experience?
Yeah, so my goal for this, and even whether or not it was academia or industry, was to absolutely pack my CV as full as I could. Like I said, I did as many presentations as I could. I tried to publish as much as I could. I did a lot of outreach. I did a lot of mentorship programs where I was mentoring students, and I tried to really just pack it out as much as I could. I think if you haven't had the opportunity to do that during a PhD, I would probably recommend trying to find an internship within a biotech company if that's what you want to do. You would instantly put yourself above some other people who have not got that experience. But yeah, for me, I think definitely getting as much on my CV as possible was beneficial for me. So yeah, that worked for me.
That's a big one. Networking is really big. Robyn mentioned Twitter. That's how Robyn got into Altos. I've used Twitter and actually even like Instagram, just connecting with people generally, right? Like you see these communities and you just like get to know them and share your personality. And even if you're not looking for a job yet, just start that relationship building now. It should be 10%, maybe 20% of your time is relationship building. Turn some of that emailing into, let's get a coffee, or let's just talk about philosophy or something else, just your personal interests. And then you have sort of warm relationships and intros. It sounds like nepotism, but it's better. And then the other thing is consider startups. Like I know I kind of keep pitching this, but like a big part of getting a PhD, I think, is marketing. Like I had to compete with people that were doing, or that had PhDs and were raising money, and I did not have a PhD. And I guarantee you it was a great marketing advantage. Like, 90% of people aren't going to look through your entire CV and read even one paper. They're just going to see that you have a CV and that you have 100 publications. And that's just like a box they check in their mind. So I think you are way more qualified to create a startup in biotech. And what do they say? A disaster is a tragedy to waste, or something like that. So, at a time when everybody's being laid off, that's cheaper labor. So if you can raise some money for a startup, then you can hire all these PhDs and do some work for yourself. If you see a gap in the market or something that you could actually do for yourself, I think it's the best time in the world to start a company. So maybe organizing some of these people together and considering that would be fun as well.
Points. I feel like Instagram is I have yet to use Instagram for research. I think that is a I aspire to someday use Instagram for research. Okay.
Not TikTok.
Well, okay. So what I wanted to ask, in kind of building off of some of these questions, there was the question earlier about skills development in your position. So I was wondering, I know at least in in postdoc positions, in graduate school positions, but also in kind of early career faculty positions, people are often assigned a mentor in their institution. And I was wondering so for example, for Robyn, when you joined Altos Labs, were you assigned a mentor, or were there people who you could go to for advice that was different than kind of a manager? And for both of you, do you have positions where you are a mentor to someone else in your current positions?
Yeah, I went to a lot of my colleagues that had already been there, so who'd been there slightly longer than me, to kind of get a feel for what they were working on and what they were interested in. When I first started, it was very much, read a lot of papers, find areas you're interested in, and start working there. And I think it was a really nice approach because it really got me into the science and into this new field really nicely, rather than just being sat there and said, okay, this is your project, your very specific area. I was given time to explore the kind of field and see where I really would fit in. So yeah, I turned to a lot of my colleagues in that case. My collaborator here, so who I report to, but he's my collaborator. Yeah, he's the one I would go to the most and see, if I have these big ideas, like we would discuss them. And yeah, I feel I was definitely, when I moved into industry, more independent in what I would want to do and what I would work on, rather than, like I said, being given that exact project. So I definitely had people to turn to when I had those ideas, but I was more independent in what I was working on.
I should have done this way more than I did. I should have reached out to other biotech founders, and I realize now that a lot of them are very happy to talk to you. But I was lucky that in all of the places that our startup was physically located, there were other startup companies next to us, and I could get a lot of questions answered that way. Awesome people and just very willing to chat and about everything. One of the greatest mentors, who is still a mentor to this day, Sam Skanga. He's an older fruit fly scientist, and I always pitch him on mathematical modeling and bioinformatics, and he's like, they tried that in the 90s and they still not getting it to work. And he's sort of the classical, and he tries to get me to read just all the classical geneticists and molecular biologists, and he's always going on about it. So he's one of the how you meet these people, I think, is the most ideal is you vibe. Like, you have shared interests and you bond just personally because you have a similar North Star. But then beyond that, if it really is just sort of a transactional thing, that can be okay as well. And I've had a few of those. But then add structure, like you reach out and you say, like, look, we have shared interest, or we have shared North Stars, even if, you don't say we don't have a shared personality, but, and then you have sort of like biweekly or monthly meetings, and it's mutually beneficial. Oh, and I do mentor plenty of people now. One of the other jobs that I had after Acorn was TKS, which is this program for youth coming up in science, technology, and startups. And I mentor too many of them. I have to say no a lot now.
Super. I do want to mention, if folks have questions that they want to ask verbally, you're also welcome to do that. You can just put a ping in the chat window, for example, if you have questions. Are there any questions now that folks want to ask verbally?
I can ask a question.
Yeah, go for it, Maria.
It's more about the idea of feeling stuck. A lot of people, will at least tell you, do not, if you move to one from academia to industry, then be careful, you're stuck. I mean, I don't really feel that way, but I wonder, is it really true? Do you feel that, once you've moved to industry, you can't get back to academia, or is it you just don't want to? Does that make sense? Like, I just feel like, is it really that one or the other? Because you do hear stories of individuals who've gone to industry and came back as PIs because that's how their journey went. But is it really a one or the other?
I really don't think it is. I think the skills that you get from being in industry will be very highly valued in academia, and I think it's more of a thing now that's coming up. And I've spoken to many of my older colleagues who are now in academia, and they've all said, like, if you go into industry for even a year or two and then you try to come back to academia, you're going to be leagues above other people who are applying for that same position, because you have a completely different experience from them, having been outside, and you have a whole new skill set that you can give to graduate students. So I think hearing that was very positive. Like, if I ever felt like going back, I've been now with Atlas just over a year, and I definitely don't want to go back. I'm really, really enjoying my time here. And I think also I'm quite lucky that I still have outside collaborations. So I have a collaboration with Alexandria, and I have some with other people. So I'm very lucky that I still have some of those outside things. So I think I still have a little sense of academia. So yeah, so I definitely don't think it's one or the other. I think if, take a year or two out and go into industry and see what it's like. And if you want to go back, I don't see why there would be any issue with that.
I'd agree. I don't have very much experience in it, but like even here, like here I am, somebody who hasn't been academia for I think 10 years now, and I'm talking to you guys, and I've talked with plenty of academics throughout even the Acorn time. And yeah, I don't know. Academics are also very open and friendly. You just gotta speak their language, and you guys already do. So I don't know. I can't say as much about this, but I can't see why not.
So I have a question from the perspective of applying for positions. So we did this mock interview program for academic positions through the SMB CDEV subgroup this year. And the most common question that I got asked for the students that I worked with was, what kind of questions should you ask when you are interviewing, right? Because any interview is going to ask you, what kind of questions do you have for us? So if you are being the person who is being interviewed for an industry position, what kind of questions would you ask the employers when they ask you, what questions do you have for us?
I still remember I definitely asked what was the kind of expectation on publications and things, because industry is different. I had no idea. So I definitely would ask, what are your expectations on your output? How do you measure your kind of goals, and how do you measure if you're successful achieving those goals? I also had questions on, if we can attend conferences. I'm part of like SMB, obviously, and some other math societies, but I'm not directly in a math position. Like, I'm in a biotech company now, but so I definitely asked if, we can still network and attend these conferences. So those were two big things for me, the big questions I had. So yeah, from what I remember, that was definitely what I asked. I can't remember anything else I asked. It was all a whirlwind, but yeah.
For the startups, I did plenty of hiring, and the best people that we interviewed were ones that asked us about our own back end, as in like, who have you raised from? How much money's left in the piggy bank? What milestones do you have to hit? I guess similar to the publishing question from Robyn, what milestones do you need to hit between now and your next funding round? So obviously that gave us a sense that they understood how startups work, even just a little bit, and the answers actually mattered to them as somebody who was about to be employed by us. Other questions around like cap table, like what's the ownership structure? Are you giving equity to new employees? And can I get some of that equity? What is your strategy there in terms of like your options pool? So yeah, asking those kind of questions, and use GPT-4 to just get the basics of what you should ask for different stages of startups, and they'll probably be really good questions.
So are there other pieces of advice that you have for students or for folks who are in academia who are considering moving to industry or to startups, or to faculty who are mentoring students? Just general pieces of advice that we haven't asked about yet that you think that we've missed that you want to share.
I don't think of anything we missed so far. I will echo what Stephen said earlier, and I think networking is a huge one. It helped me get my position. It helped me get my visa for the US, oddly. So yeah, that was a massive one for me, was networking. And I've said this again, but like, presentation skills. You need to be able to present to many different kinds of people. So get as much experience as you can with it, and be comfortable with presenting. I spent a long time practicing, owning the silence in presentations, not filling it with needless words and things. And so yeah, I think that's two big things for me that have definitely helped me in my career so far. Yeah, maybe I'll think of something else, but that's what I have.
For me, it's more just like an encouragement. I hope more academics can transition into like startups. So computer science had its moment when basically everybody in software and computer science could very easily create startups and like succeed, and then even like give back to academia and sort of have like an incestuous back and forth relationship. And I'm really hoping that in this next 10, 20 years, it's going to be like the age for biology. And I do see more software people kind of making the move over because there's more to work on, and it's very relevant because we are biology. Humans are biology. So I'd encourage more people to look at the business world a little bit, just study Silicon Valley and understand how these computer science people went on to create new technologically interesting science-ish based companies with a moat that allowed them to prosper in ways that were, significant and went beyond like the norms of academia in interesting ways. It's a cool and interesting life that is available that I think is something that I'd encourage more academics to try out, and just talk to me personally if you're curious about more details on that. So more encouragement than specific advice.
Super. All right, my last question, but first, are there any other questions from audience members before it?
Can I just make a comment more
Go for it, Maria.
the startup? It's more about grounding it. A lot of, let's say, researchers, postdocs specifically, or we're poor, or at least a big chunk of us. So I know I really love the idea of being encouraged to go into a startup, but how do you do it if you're barely like surviving? What you have pennies, and so you have 2 kids and it's a multiple body problem. Like, I just feel like, what do? You have to live on the streets while you're building your startup. Like, it seems to be I love the encouragement, but I want to know, like, how would somebody in these kind of situations where we're most of the time underpaid, barely scraping by, gonna jump to that kind of world?
Totally. So yeah, I was very fortunate that in like third year we had $400 a month rent and like $200 for food, and we had like $300 left over at our $1,000 a month salary to ourselves, and we could afford to do that for 3 years. So that, obviously a very different situation. We roommated together and stuff. Now it is a little different. So the average age of successful entrepreneurs is actually in the 40s, not in 20s, which says something. That is a little bit interesting. So, obviously everybody's going to be a little different. There's the sort of raising friends and family round. So different people, literally just based on their personal situation, how many months could you accrue from your friends and family so that you can work on this full time for even just 4 months? You take a break from what you're doing in academia, maybe between your PhD and your postdoc, or your PhD in industry, and you just spend 4 months just giving it a freaking shot. And then at the end of the 4 months, you've run out of your personal runway, you and a friend or 3 friends, and then you call it a day and you move on. But it honestly is an incredible thing on your resume anyway, that you explored this very thoroughly. So that's one piece of thinking. Another one is just the spare time that you have. I don't know, like, if you're very passionate about an idea and you feel like you're onto something and it really could become a unicorn kind of thing, then, trying your best to talk to your PI or whoever your boss is and saying like, hey, look, I had this really interesting idea. I'd love to be able to spend 10 hours a week on it, 15 hours a week on it. And if you do that over 4 or 5 months, you can build up a real serious body of work. And then as long as you have 1 or 2 other people, 3 people, 4 people, you pitch that to VCs and you develop these relationships over time. And then before you know it, boom, here's at least 100K in a sort of pre-seed that you can use to explore. More and more people are getting kind of funded that way. There's also pitch competitions you can apply to. Like, literally, they'll just give you 50K, just like that. And now you've got a piggy bank with 50K, and you can explore your idea that way. So it's surprisingly doable, but I can also see why it's scary, Maria, and I can see how it could be more complicated than that. But yeah, I hope that answers the question.
No, that was great. I just thought, to move to that, I think you gave a good ideas. Pitch competitions, all of those are accessible, but it's nice to know that they are out there.
All right. Well, my last question is if you could share what you are research or scientifically most excited about right now, and also your favorite part of your job.
Yeah, so I won't say much explicitly what I'm working on right now, but I am most excited about collaborating with experimentalists. So we have more experimentalists with Altos than computationalists, and it is so great to just go up into the lab and say, I really need this parameter for my model. Can we run some experiments for this, please? And generally, yes, we can. It's a really, really special environment, and I'm very, very happy and lucky to have kind of access to that sort of thing. So yeah, I'm working on, lots of different models theoretically, and then I try and validate them experimentally. So it's a very nice kind of workplace to be in. And I get to travel quite a lot with my work. We have institutes in the UK and also another one in California. So I do get to visit them often. So it's something I really enjoy doing, is traveling and getting to meet with other groups and meet with other collaborators that we have. And so yeah, it's something I particularly enjoy about the job.
What was the question again? Sorry.
Research or scientifically, what are you the most excited about right now? And also just what is your favorite part about what you're doing right now?
Yeah, fair. Thank you. Sorry. So I'm very excited by the idea that next generation LLMs will allow us to understand biology and produce outcomes that are very unintuitive. Like you look at like quantum and many other fields. Human brains are sort of like formed in such a way that they're going to come up with certain theses and hypotheses. And I'm excited by the idea that if AI can understand enough about sort of like biology from a systems level, it may unveil some low-hanging fruit that we have not been thinking about that is just outside of the grasp. So I'm really excited by that. It's blue sky thinking, and it might not be possible. And it might be that like it will just uncover nothing, or biology is way too complex and it's going to require GPT-15 level of compute or something. But I'm excited to just see it as every layer kind of keeps getting unfolded, whether we'll come up with discoveries that we never thought would be possible. So that's what I'm spending all my time. And then I'm on sabbatical right now, which means that I don't have a boss. I wake up at 10:30 every morning and I just go to the coffee shops and I just kind of program and do my thing. And I spend a lot of time playing music right now. So I have a very kind of free life. Honestly, it's quite nice. But I also see you guys learning a lot and having real pressure. And I feel bad that I am not good enough at putting harder pressure on myself. The number of textbooks that I've bought, I have like 3 embryology textbooks and at least 2 AI textbooks, which I have allowed to collect some dust because there's nobody who's going to, put me through the paces and say, Stephen, you have to learn this in order to, be useful. So a little bit of self-initiative, even when I'm sick right now, you guys will push through it because you have people all around you that are depending on you. Nobody's depending on me right now, except for myself and my future self. So it's a two-edged sword. So yeah.
I think breaks are good too. Well, all right. Well, I think that is all of our questions. Thank you so much, Robyn and Stephen. This was really lovely. Thank you for taking time to chat with us and answering the questions. And yeah, we appreciate it. Let's give them a round of applause. Thank you both.
Thank you.
Thank you guys for putting this on.